Hello and good morning, everyone. So we are continuing with our topic on regulation of gene expression. But before I start this topic, just a few things. You might have seen your grades for assignment. If you haven't, those are mostly the ones who directly submitted their assignments to me because you couldn't, for some reason, submit it on Web Campus. So I will grade those separately. So if you don't see your grades by Friday, shoot me an email. Okay. Other than that, I received a few emails about the answers on test 3. I will send you a key so everybody can see the correct answers, and also I'm working on the emails, so I will send you the replies back as soon as I can. Okay, and with that, so today we will be talking about regulation of gene expression by regulatory RNAs or non-coding RNAs. The topics that we are going to cover today are gene regulation by regulatory RNAs in prokaryotes as well as eukaryotes. So we will be talking about CRISPRs, RNAi, short interference RNA, miRNA, microRNAs, and all that. This is mostly from 7th edition of Watson, chapter 20. The page numbers are noted here. In addition to those page numbers, you might want to read one or potentially both of these papers. These are really good papers to give you a lot of background about this topic. And with that, let's move on. Okay. So regulatory RNAs, so these are the RNAs that are not translated, so they don't make proteins, but they are produced from RNA pol II. These are also called non-coding RNAs, which is you probably have heard a lot more, because that's the term we usually use for the RNAs that do not translate. So these are non-coding, these do not code for a protein, but they have regulatory functions. For a long time, scientists thought that these were just like junk DNA. These didn't have any function. But more and more research is showing that there are more families of these non-coding RNAs, and also these non-coding RNAs might have, they do have regulatory functions. They might work by different mechanisms. So their basic role is in gene regulation in all organisms, in prokaryotes as well as eukaryotes. As you might have seen, this is the theme throughout the living systems, that eukaryotic systems are usually more diverse and complex than prokaryotic system because the genome is much larger in eukaryotes compared to prokaryotes. But the basic mechanisms of most things is very similar in prokaryotes and eukaryotes, and that provides a really good way to study different mechanisms by using these simpler organisms, for example, a bacteria. These non-coding RNAs, the way they work in all cases is by hybridizing to a template, to a DNA sequence that is complementary to that non-coding RNA. Usually they reduce expression of the target gene, but they can also activate the expression of the target gene. So here in this example, you can see these two different examples here. One is activation, one is repression. So here you have a ribosome binding site right here, and this is coding sequence. So this is the DNA sequence that will code for an mRNA, right? And here, if this is an activation mechanism, so this ribosomal binding site is not available for the ribosome to bind. So this will be this is your mRNA. So this is the coding sequence for that mRNA, and this is ribosomal binding site, but this is not available for the ribosome to bind because this 5' end is bound to this ribosomal binding site. So it makes this loop, and the ribosome cannot bind. In activation, these small RNAs, they can pair with this sequence and unmask the ribosomal binding site. So here, this sRNA has the sequence similarity to the 5' end of the mRNA. It just binds to this sequence. Once this binds, it's binding of this molecule will change the sequence, change the conformation, and this change in conformation will then make this RBS available here, and then this is activated. Here in this example, this is an example of repression. This is ribosomal binding site. This ribosome will come and bind here and will lead to translation. But if this sRNA binds to this has complementary to the sequence, the ribosomal binding site sequence. If it pairs with it, or if it binds to the sequence, now the ribosomal binding site is not available for the ribosome to bind, and that will lead to repression. So in prokaryotes, the small RNA riboswitches, or CRISPR, are the three different mechanisms by which these regulatory RNAs can change the expression of different genes. The small RNAs, we just talked about the example of small RNA, that it can activate the expression or it can repress the expression. And when we say expression, that means the synthesis of protein, right? So these small RNAs are about 80 to 120 nucleotide long. They are produced from their own genes without further processing. So these are about 100 small RNAs in E. coli. These produced from their own genes. Most small RNAs have weak complementarity to their target mRNA. That means they don't have to be exact match. They can very well tolerate the mismatches in the sequence. So this is these are a couple of different mechanisms that it shows. We will go one by one with this. So these small RNAs, they bind to this RNA binding protein, which is a chaperone. It's called HFQ or HF1. This HF1 is the gene. So they bind to this RNA binding protein. This will stabilize the RNA, and this will help the RNA to hydrogen bond with the target. So here, this is your this is the mRNA. This is ribosome 30S subunit. So this is the mRNA is ready for translation. This sRNA, this short RNA, small RNA, this is bound to this HFQ, the protein, the chaperone protein that brings it there. And it's bound to this, and this complex, this ribonuclear protein complex, RNP, then comes here. Finds the target sequence, somewhat complementary sequence of this sRNA on this mRNA. Right? This will then bind to that sequence. Okay. So this entire structure, that means this sRNA HFQ, this RNP, this becomes a substrate for RNase E. So here's RNase E. RNase E is an endonuclease, which means it can break down the complex. So this RNase E here, so this comes and bind here. This is bound here to this mRNA. So it's found the complementary sequence, bound to the complementary sequence RNase E, then will break down this this hybrid here, this mRNA sRNA hybrid. Okay. This because it cleaves this target mRNA, and if the target mRNA is not present there, that will lead to reduced gene expression. So that's the main mechanism of siRNA, as RNAi as well, that the degradation of mRNA will lead to lower gene expression. Some siRNAs, some of these small RNAs, they can bind directly to their mRNA complements and can activate or repress translation by altering the sequence or exposure of Shine-Dalgarno sequence. And you remember, Shine-Dalgarno sequence is important for binding of mRNA to ribosome. If the ribosome cannot find Shine-Dalgarno sequence, it will not bind to mRNA, so it can repress translation by altering the exposure to Shine-Dalgarno sequence. Okay. So here in this example, this is your mRNA, this bound to 30S ribosomal subunit. This sRNA can bind to this subunit and can degrade it, or will not allow it, since it's bound there, it will not allow the whole ribosomal assembly to happen. That means mRNA will not be able to translate. That's the inhibition of translation. In activation, this is the RBS, ribosomal binding site, ribosomal binding sequence, and this, as we saw in the previous example, this sequence, the mRNA 5' sequence, bound to this RBS. So in order for this RBS to open and ribosome to bind here, this need to be uncomplexed from here first, and binding of this sRNA can open the sequence, and this will allow the activation, allow the synthesis of protein here. Production of sRNA from ribonuclease cleavage. So here, this is RNase E. If the sRNA is without the complex, without the chaperone, then it will be broken down by RNase E. But with the chaperone, it gives us this stability. So this chaperone makes it stable. This figure shows the induction of ribonuclease cleavage of sRNA and mRNA. Once the sRNA binds to mRNA, it makes this double-stranded molecule here. This will activate RNase E activity, and it will break down both, not just the sRNA, but sRNA-mRNA complex. And since this is the degradation of mRNA, this mRNA will not be able to make protein. Another mechanism by which these regulatory RNAs regulate gene expression in prokaryotes is called riboswitches. These riboswitches are specific sequences, so we'll see. These are typically found on the 5' UTR of the genes they regulate. So these are cis elements. They're made of two components, aptamer and expression platform. So here, this is this in green is a riboswitch. This is the aptamer. So this is this molecule is just bound onto itself, right? So all these are wherever this mRNA, this sequence can bind onto itself. This will make these loops and these hairpins, and this is the expression platform. It's another part of the riboswitch. So riboswitch has two components: this aptamer and this expression platform. This is right before the ribosomal binding sequence here. So this is on the upstream of the translation start site. So it's upstream of ribosomal binding site on the 5' end. These aptamers, they can bind to small molecules. So if the small molecule bind to the aptamer, that will result in conformational changes. That means it will change the sequence of this, or change the conformation of expression platform. So right now, if there is no small molecule bound here, expression platform is not inhibiting a ribosomal binding site. Ribosome will come and bind to ribosomal binding site, and it will the translation will continue, will start as usual. However, if the small RNA, if the small molecules they bind to this aptamer, then this aptamer will the conformation of aptamer, the binding of this small molecule to aptamer will change the structure of expression platform, and the change in structure of expression platform will now inhibit the binding of ribosome to ribosomal binding site, because this is not available for the ribosome to bind. So this is how riboswitches work, and this is how riboswitches can regulate expression of gene. So now nothing changed. It's just the aptamer this sequence was already present. If there is no small molecule binds here, everything will go as usual. But this binding leads to this change in conformation that will now inhibit the binding of ribosome. That means there will be no translation. Now, third mechanism in prokaryotes for regulation of gene expression by by RNA is CRISPR, and we already talked about this quite a bit in the class, but I will just talk about it again, so this will be refreshed in your memory. CRISPR in E. coli is used as a defense mechanism against bacteriophages. It can be plasmid, it can be transposable elements. So any of these can be broken down using the CRISPR mechanism. Mostly it's used against phage, so that's the best studied example mechanism is how bacteria protect itself from infection of these bacteriophages or viruses. In a way, it works like antibodies record of infection. So you're probably hearing a lot about immune response, immunization, antibody production, herd immunity, and all that right now. That's how this antibodies, the previous infection can lead to a more robust response during next infection. It provides this immunity. So this is what the bacteria is doing as well. So CRISPR is short for clustered regularly interspaced short palindromic repeat sequences, and the reason for that is because the CRISPR in bacterial genome there are these sequences are clustered in one place. These are regularly interspaced because there is a repeat spacer. And these are palindromic these repeat sequences are palindromic. So that's where the name comes from. These regulatory CRISPR RNA or these ncRNA, CRISPR RNA, they promote degradation of the invading genome, so usually the viral genome. CRISPR can also promote degradation of mRNA produced by the invading genome. So if it's a viral genome, it can degrade those genome as well. But one thing to keep in mind, and I'm going to spend at least a half a lecture on retroviruses on either Friday or Wednesday or Friday, because this is all to put it all in context with what we are dealing with, SARS-CoV-2 now, which is an RNA virus and a retrovirus. So keep in mind that most bacteriophages are DNA viruses. Okay, so this mechanism is more suited for these DNA viruses. Okay, so keep that in mind. Okay, so this image here just it shows that how this CRISPR can be used as a tool, or how we are using it as a tool. So I will not spend a whole lot of time on this right now, but we will talk about this in maybe later. So this is so we depending on what we know how CRISPR works in bacteria, we can design a similar, or we do design a similar guide RNA. This guide is the guide has only these 29 nucleotides that's complementary to the sequence that we want to add it. In addition to that, there is a linker loop and tracer RNA. So this is the linker loop, tracer RNA, and CRISPR RNA. This CRISPR RNA, this is the regulatory this regulatory RNA. It's these CRISPR and tracer RNAs. These are the same sequences. These really come from this repeat sequence of the CRISPR of bacteria. So the spacer is the unique sequence, but this repeat, these are the same sequence over and over. So that's why this CRISPR tracer RNA, this is all the same sequence for all of your guide RNAs that we are synthesizing. This provides stability to this portion that will need to find the complementary sequence. Okay. These repeats are about 30 nucleotide in bacteria. It's usually about 30 nucleotide and also separated by approximately 30 nucleotide of spacer sequences. These spacer sequences are various, they come from various pathogens. That's why spacer is unique. But the repeat sequences are the same sequence, same palindromic sequence over and over. These various pathogens can be a phage. Most of the time it's a phage. It can be a plasmid because the bacteria doesn't always want to have these extra nuclear molecules, these plasmids, or transposable elements, because transposable elements can be really harmful for the genome. the DNA transposable elements for sure can hurt the genome a lot, and the retrotransposable element, because they are very similar to retroviruses, RNA viruses. These can be also identified by these mechanisms. These are preceded with a 500 nucleotide A plus AT rich leader sequence, and you will see why this leader sequence is important, because this leader sequence has its own promoter. So it has a Tata box there. Various CRISPR associated sequences or Cas sequences are encoded. These are enzymes. These are the nucleases. These nucleases work along with the CRISPR to make the double-stranded break. These Cas enzymes there are several of these Cas enzymes are identified: Cas1, Cas7, Cas9, Cas12. They all can excise a portion of pathogen's DNA and insert it into CRISPR locus. Cas12 and Cas13 are specifically for RNA, so they can cut down that RNA sequence and use that to add into the CRISPR by first converting into DNA. This can excise a portion of the pathogen's DNA and insert into CRISPR locus close to the leader sequence. Okay, so this is the leader sequence, that AT-rich sequence, about 500 nucleotide, and then each of these sequences is all these gray are repeat, and all these different colors are spacer sequence, because these spacers are the unique sequence. When a new sequence is entered into or loaded onto this CRISPR region, this usually goes here close to the leader sequence. So the newer sequences will be closer to the leader sequence. The older sequences will be away from the leader sequence. How it works, let's see. So this is a bacteriophage. This bacteriophage is it's inserted its DNA. It's injected its DNA into the bacteria. So this is a foreign DNA. Now this foreign DNA, this is the sequence of foreign DNA. This foreign DNA it will be recognized by the cell as a foreign molecule, and this the cell then makes these proteins, these Cas proteins, and these Cas proteins here, Cas1, Cas2, probably some other factors that come together, and these will make this this break into this sequence. If the sequence is not already present in their CRISPR cassette, then they will make this little break, this sequence. They will cut this sequence and put it into this bacterial genome at the CRISPR site. So they made this break. This is the PAM sequence, and make these Cas9. So Cas usually recognize 3 to 5 nucleotide sequence. These 3 to 5 nucleotide sequence are different for different Cas. For Cas9 it's NGG, but for Cas1, Cas2 this will be different. So these will find that sequence and will make about 30 nucleotide cut in the foreign DNA at that site. It will take this sequence, and then it will make the break here in the CRISPR cassette. So this is the leader sequence. This makes the break right here. This will be inserted. Now here is the new spacer inserted here, and then the repair mechanism will fix on both sides of this repeat. So this repeat is now one repeat becomes 2 repeats, because it was cut right here at the end of the repeat, between repeat and spacer. So this will be the 5' spacer of this this repeat sequence here, and this will be 5' spacer of this new 5' repeat of this new spacer. So here's your new spacer, repeat, spacer, repeat. Okay, so this is bacterial DNA. Now let's think about a scenario where this CRISPR, this bacteriophage has already infected the DNA, the bacterial genome. So bacteria already knows about this the sequence. So the sequence is already in the CRISPR cassette. So here the bacteriophage infects the bacteria. Bacteria makes this whole sequence. Bacteria will make this, will synthesize this whole cassette. These will be broken down into individual spacers. These individual spacers will be loaded onto this cascade. It will go and look for the sequence similarity. It found the sequence similarity, and then it will broke down this bacteriophage DNA. So it will be cut down here, and that way this bacteria can help itself not getting infected. Okay. The mechanism, as the leader has a promoter, so you might have noticed here there's a Tata box here in the leader sequence. So this Tata box is that's where the promoter is. This promoter, it has a promoter that it allows the mRNA containing all of the repeats and spacers to be produced. So when this makes this mRNA, it will make this all of these sequences. So it will copy the whole sequence. So here. The Tata box, the transcription will start from all these transcription factors will come and bind to Tata box. Transcription will start. It will transcribe the whole cassette. Okay. Here. So this is called pre-CRISPR RNA. This pre-CRISPR RNA is all of the repeats and spacers together. So each of these sequences, these 3' handles are the repeats. Okay, so this is pre-CRISPR RNA. This pre-CRISPR RNA, then it makes this cascade complex with Cas proteins. So this is a cascade complex. So it's associate itself with one of the Cas proteins. So here is the example is Cas6. So here's the example is Cas3. So whatever Cas it's using, so different bacteria have different bacteria make these different Cas proteins. So these Cas proteins will be associated with the Cas proteins. Now this cascade, this enzyme complex, this Cas encoded proteins, they form an enzyme complex called a cascade. It binds to pre-CRISPR RNA. And digest it to form individual CRISPR RNA. So this binds to this pre-CRISPR RNA. It will break it into individual CRISPR RNA. Now each individual CRISPR RNA will have a short sequence from the 5' end and the complete 3' sequence of the repeat sequence. So each of these will have a short 5' and most of the 3' sequence. So this will give it this stability here. Okay. So here shows this binding with this cascade complex. This binds with the Cas, and along with Cas, it will make this break at the sequence that's complementary to it. So same here, makes this complex. Once the cascade complex cuts it into small individual pieces, these individual pieces then bind to this Cas. This whole complex will find the sequence that's similar to a target RNA, and it will make these cuts. So here in this example, it's the 5' handle, that's what's helping with stability. Here this is 3' handle. So either 3' or 5' handle can help with this, provide the stability to the sequence here, to the spacer sequence. This cascade promotes association of mature CRISPR RNA with its cognate genomic DNA. So the cognate genomic DNA will be the genomic DNA that has sequence similarity to that sequence. From the pathogen, it will cleave the genomic DNA at the hybridization site, and that will then lead to degradation of that sequence. Now, protospacer, we talked about this is the protospacer region. This is the sequence of the phage that get inserted into CRISPR. So this sequence from here to here, PAM is the protospacer adjacent motif. This is the sequence that is recognized by the Cas enzyme. So this is downstream of protospacer, and so that's where the Cas will bind and make a cut. Okay. precision engineering, use CRISPR using as a tool in molecular biology, has also has the same component. So now we are not talking about regulation of gene expression in a system by its own regulatory sequences, but this is more of using as a tool. So we used Cas9 as the most widely used enzyme. This is an endonuclease that cuts DNA. For your guide RNA, you will design this small sequence, about 20 base pair, 20 to 30 base pair, usually 20 base pair. That's complementary to your sequence. And then this is the CRISPR RNA, that CRISPR tracer RNA together, that is there for providing stability. This is the same sequence for everything. Now this your guide RNA is whatever organism you want to add it, you will make the sequence similar to that gene. So we have already talked about it. These two things come together. It finds this guide RNA will find its cognate genomic DNA sequence, will bind to that genomic DNA, and binding of this cognate guide RNA to its cognate genomic DNA will then bring the Cas9 there, and it will make the double-stranded break. These double-stranded breaks will then be fixed by the cell's mechanism, cell repair mechanism, and we talked about there are two main repair mechanisms: homology-directed repair or non-homologous end joining. So either of those will be employed at this point. Precision engineering using HDR, homology-directed repair here. So as we know that there are each. Let's go back. The genome is broken, it must be repaired. If it's broken on one chromosome, there is another chromosome. That chromosome, it can use that chromosome as an example to fix it. So that's what recombination is like. So it can use this. It will recombine with the chromosome that's intact and will copy that sequence. We can use that mechanism to insert a new sequence if we provide a synthetic DNA molecule. So that's that synthetic DNA molecule. This is what we want to insert here. So we will use this homology arm, which is similar to these sequences. So these homology arm will be recognized here, and because these are similar, it will assume that this sequence is the one that's missing. So this can be repaired by this donor DNA that we have provided. Okay. So that's how we use it as a tool. Okay. And now, this is a nice video here. I'm not going to play it, but I would recommend that you watch it. It's a really nice way of showing how the CRISPR works. So now, moving on to regulatory RNAs in eukaryotes. As we all know that the RNAs regulate gene expression in eukaryotes, and you probably worked on a number of papers that have used microRNAs as regulatory sequences, regulatory RNAs to study the gene expression and how these microRNAs affect the gene expression. You might have worked on some of the papers that use RNAi, RNA interference mechanism to change the gene expression. So these are very common in eukaryotes. 30 to 70% of genes in higher eukaryotes are regulated by RNAs, so it's a very common mechanism. These RNAs can be short, so short interference RNA or siRNAs. This can be microRNAs or miRNA, pvRNA or piRNA, or these could be long, simple long non-coding RNAs. RNA interference or RNAi is a post-transcriptional gene silencing mechanism, and it's a very important research tool. And this is based on the cell's own mechanism of to deal with plants. A lot of animals use it. Again, the eukaryotes use it as a way to break down viral genomes, viral DNA, and also to regulate their own expression as well. The control of gene expression is caused by caused by the presence of double-stranded RNA inserted in the cell. It leads to loss of mRNA with homologous sequence, but that's how you use it as a tool. You can use, you can design a double-stranded RNA or an siRNA that has the sequence similarity, exact sequence similarity to the mRNA that you want to break down. You insert it into the cell in any way. Either you soak the cells with the siRNA solution, you can directly inject into, you can electroporate. So there are a number of different ways you can get that siRNA or double-stranded RNA into the cell and then let the cell's RNAi mechanism do its job. So because the RNA interference mechanism in cell is mostly there to break down foreign DNA, so if you add something, that will be considered as a foreign as well. Complete mechanism of RNAi or siRNA, how siRNA, miRNA, or long-coding RNA work is not fully understood, but what we know is that's what we're going to talk about today. Double-stranded RNA, if you inject it into a cell, so now we are talking about double-stranded RNA as a tool. So if you inject it into a cell, the specific nucleases, for example, dicer, will digest this double-stranded RNA into 21 to 23 nucleotide long fragments. So these are called now siRNA or short interference RNA. You can design these 21 to 23 nucleotide long fragments directly and inject, so that way you don't have to rely on the dicer to break down the double-stranded RNA. So either you inject siRNA or you inject double-stranded RNA that will be broken down into siRNA. Once these siRNAs are there, these bind to the RNA-induced silencing complex, or called RISC complex. So this is a RNA protein complex now. It's a multi-subunit protein complex. It includes argonaute or AGO, which is an RNase. Okay. So once the RISC complex, this RNA-induced silencing complex or RISC has a single-stranded RNA, it is considered as an activated RISC complex. Okay. This, as I said, is likely evolved as a defense against transposition or viral infection. That's what plants usually use it for, and animals also use it for the same mechanism, same defense mechanism. There are two ways of gene silencing that's caused by the siRNAs. So triggering mRNA degradation. So in this, your mRNA will be degraded. This is the most common mechanism as far as we know. In this mechanism, RISC, RNA-induced silencing complex, is hybridized to the target mRNA via its siRNA. So the sequence similarity will lead to the binding of siRNA to mRNA, and siRNA bound to mRNA will then be recognized by the proteins, for example, AGO, and this will break down those double-stranded RNAs. Here, so this is a long double-stranded RNA. If you inject this long double-stranded RNA, or if an organism, say for example a virus, injects this into the cell, this double-stranded RNA will be broken down into smaller pieces by this nuclease dicer. This dicer cleaves this double-stranded RNA, it's long double-stranded RNA. So think about DNA viruses. So DNA viruses are double-stranded, usually double-stranded DNA. So these double-stranded DNA molecules will be inserted into the cell. These will be broken down by dicer into small pieces. These small pieces, siRNAs now, they make this complex with RISC. This is an activated RISC complex. This activated RISC complex will find the RNA, mRNA that has the same sequence, complementary sequence to this siRNA. This siRNA will then bind to this. This siRNA will bind to the sequence that has complementarity. This complementarity, the sequence that has this complementarity, will then be broken down. So this This mechanism here with RDRP, how it's elongate siRNA, we will talk about that in a minute. Okay. So the binding activates Argonaute to digest the mRNA. Now that leads to the slicer activity. So once it's cleaved, the mRNA fragments are rapidly degraded and this will not be able to make protein. This siRNA complex can also be amplified to make long double-stranded RNA again. So remember, this long double-stranded RNA is cleaved into small pieces, but these small pieces can again be made into longer double-stranded RNA. This can be achieved by RNA-dependent RNA polymerase. So this is a different kind of polymerase. This is not a DNA-dependent RNA polymerase. It's not RNA-dependent DNA polymerase. So it's neither one of the polymerase that is used for transcription. It's not a polymerase that will be used. It's not a reverse transcriptase either. It's a different kind of polymerase. This is an RNA-dependent RNA polymerase. So this will use RNA as a template to make this RNA molecule. So it will elongate it, and this elongated double-stranded RNA that can be cut again into siRNA. So here you can see that one molecule of this long double-stranded RNA can be made into siRNAs and then again make into double-stranded RNA into siRNA. So just one molecule can break down all of the mRNA cognate to that siRNA. These RNA-dependent RNA polymerases are very are common in RNA viruses or retroviruses, and I would like to spend at least half of a lecture later in the week talking about not only about the RDRPs but the RNA viruses in general, and how these RDRPs are very important for viral genomes, and how that's how we're using these RDRPs, target these RDRPs in order to make a drug against the virus, so antivirals. So this RDRP can, because it extends the siRNA in the activated risk complex before slicer activity, so it can make its multiples of these siRNAs again. So this creates a long double-stranded RNA that becomes a substrate for dicer to create more risk in the cell. This siRNA signal can be amplified because of the activity of RDRP. If the RDRP is there in the genomes, then this can amplify the siRNA signal. There's no RDRP found in humans. RDRP is present not only in viruses but in also some of the eukaryotes, but not in humans. And I will go over this later when we talk about retroviruses. So that was the first mechanism was triggering mRNA degradation. That mRNA can be degraded. Then another way of regulating gene expression by siRNAs, by RNAi, is reducing transcription. So in this, instead of breaking down the already synthesized mRNA, we can also reduce the transcription of that gene. And that's by changing the histone proteins, changing the chromatin structure of the DNA. So reducing transcription is when a double-stranded RNA associates with Argonaute 1 in a RITS complex. So this is RNA-induced initiator of transcriptional gene silencing complex. So here, this is double-stranded RNA. This double-stranded RNA dicer cleaves it into siRNAs. So this can, again, if there is an RDRP enzyme, it can amplify into double-stranded RNA, again broken down into siRNA again, in presence of RDRP only though. So in this is double-stranded RNA, dicer cleaves it into small molecules, small siRNA, 21 to 23 nucleotide. These siRNAs then complex themselves with Argonaute. This Argonaute will complex with other proteins, making this RITS complex, this RNA-induced initiator of transcriptional gene silencing complex. And this Argonaute, along with siRNA, along with RITS complex, will bind to the sequence here to the complementary sequence of that siRNA. This binding will then lead to initiation or lead to these different factors. So this siRNA enriches associates with complementary sequence on the DNA. Here it recruits a methyltransferase that will methylate the histone H3. Remember, there are H1A, H2A, H2B, H3, and H4. There are 4 of these histones in dimer, make an octamer of the nucleosome. And as we also remember that H3 and H4 sequences are very conserved, but these can be changed by adding these different groups on their tails. So in this case, it adds methyl groups on histone H3. This methylated histone H3 then attracts this SWI16. This promotes heterochromatin formation. So here, this promotes heterochromatin formation. This heterochromatin formation then spreads, and this because it's now heterochromatized, or this is very tightly bound, it's not available for the transcription machinery to bind to this DNA sequence, and that way it will reduce the transcription. It will decrease because this is not available for transcription, so there won't be enough transcription of this gene. This mechanism is conserved in mammals. They use different control regions. For example, promoters or enhancers are usually targeted by the Ritz complex. Coding sequences aren't, because this is the inhibition of transcription. So if the promoter is not available, then the transcription won't be able to continue. So that's why it's more important to target for the Ritz to target those regulatory sequences. MicroRNAs or miRNAs, they have similar functions to small interference RNA or siRNAs. And these are different class of regulatory non-coding RNAs. So we talked about siRNAs. Now we are talking about miRNAs or microRNAs. MicroRNAs are larger than siRNAs, because siRNA is, double-stranded RNA can be large, but the siRNA itself will be only 21 to 23 nucleotide. MicroRNAs, when these are they aren't the mature microRNA, so as such the microRNA is around 75 base pair long, 75 nucleotide long. However, the activated mature microRNA will still be small, similar to that of siRNAs. So here, this is microRNA is produced from a 75 nucleotide stem loop RNA. So this is the pre-microRNA. This is microRNA gene. So the microRNA, their own gene will produce these microRNAs. So the microRNA gene will transcribe into this primary microRNA or pre-microRNA. This pre-microRNA has the stem loop RNA. It has this loop here, and this is the stem. This is one of the cleavage site. So this pre-microRNA will be cleaved here by one of the nuclease called DROSHA. DROSHA makes this cut here and here, that this makes it a pre-microRNA. So instead of this primary microRNA, now it's called pre-microRNA, because it's not the mature microRNA yet, because it still has that structure here, the loop here. So these 75 nucleotide stem loop RNA structure, these are broken into smaller pieces that will make a RISC complex by dicer, similar to what we saw in the siRNA as well. So dicer will cut it into the double-stranded RNA into small pieces. In microRNA, there are 2 steps. There's DROSHA cuts the stem first, and then the dicer will cut into small pieces that will then associate themselves with RISC. So this cleavage happens into nucleus. So in the nucleus, Drosha cleaves the stem loop structure here. The rest of the transcript that forms this pre-microRNA then exported into the cytoplasm. So here it's in the cytoplasm. This again here, it shows that this is the basal sequence, lower stem. This is the site where cleavage by Drosha will happen. It's the +1 site. So right here, mature microRNA is about 22 base pair. It's right here, middle, and this is the site where dicer will cleave itself. So cleave this microRNA. This, the terminal loop will be then removed, and this will be an activated microRNA or mature microRNA. So this is mature microRNA. Mature microRNA here, once dicer makes this cut here, cleaves this terminal loop, then this sequence is microRNA. Duplex sequence will since only one of the sequences is required, it's needed in the risk formation, in the risk. So that's one of the sequence will be degraded. Second one, the 3 to 5 prime one, will associate. This will be degraded because this is not required now. This won't make the complementary sequence. This 5 to 3 prime sequence that will then go with risk formation with agonauts. And this mature microRNA then can work in 3 ways: mRNA target cleavage, transcriptional repression, or mRNA de-adenylation. So either way, it will then affect the gene expression. The stem loop pre-microRNA is exported to the cytoplasm, where dicer removes the loops. Remove this loop here. That translational silencing requires this mature mRNA, which is 18 to 25 nucleotides. So it can be a little bit bigger than siRNAs, but in the same range-ish. And these microRNAs usually pair with the 3' UTR of target mRNA. And if you recall, when we were talking about the 3' UTR of target miRNA, when the ribosome assembly happens on this mRNA, it's in eukaryotes. It's not only binds to the 5' region of the mRNA, but it also binds to the 3' end of the mRNA to keep it in the loop. However, the binding of these microRNAs, it can change that regulation, so it can lead to translational repression, because it's usually interfere with the translation. Most microRNA base pair imperfectly, so they don't have to be exact a match of their target mRNA, but as long as they can bind, they have enough sequence similarity to their mRNA, they can bind to this 3' UTR of their target mRNA. They usually do not cause mRNA degradation if they are not perfect match, but they can still inhibit translation or repress translation. To cause mRNA degradation, it need to be a near perfect match, if not perfect. MicroRNAs are important for development and environmental response, and you probably read that in multiple papers already. About 75,000 microRNA have been identified in the model plant Rhabdopsis, around 150,000 in rice, and several thousand in humans. So they appear to have very important function. One of the things to remember with microRNAs is not a one microRNA, one mRNA regulation. There can be multiple microRNA that regulate the same mRNA, and there could be one microRNA that can regulate multiple mRNAs. So the regulation by microRNAs is very complex, and we still don't know how many microRNA might be regulating expression of one gene, or how many genes will be regulated by one microRNA. So just to summarize it again, this is the siRNA. The mechanism: long double-stranded RNA is cleaved by dicer into small interference RNA. These small interference RNA will then make a risk complex. This risk complex, because there are now two strands of RNA. One strand will be cleaved, another strand, the guide strand, will associated with this risk complex. AGO is very important, this nuclease. So AGO will bring this guide RNA to the risk complex, and then this siRNA will recognize the target. This target recognition then can lead to mRNA cleavage, DNA methylation, translation inhibition. siRNA can be amplified into double-stranded RNA and fed back to the risk complex by the RDRP, or RNA-dependent RNA polymerase. So siRNAs versus microRNAs. So these siRNAs or microRNA, either one of these, they work and somewhat similar, but different, because microRNA needs to be cleaved twice, once in nucleus by DROSHA, then in by dicer in cytoplasm. siRNA also converted into these small interference RNA. The double-stranded RNA will be cut by dicer into these shorter fragments, 21 to 23 nucleotide, 18 to 25 nucleotide in microRNA. These will make this complex well argonaute, either of these. And then it leads to two different ways: RITS, or risk complex. So RITS is RNA-induced initiator of transcriptional gene silencing that will lead to DNA or histone methylation. So this DNA is not available to transcribe now. So that will be a transcriptional silencing. So no mRNA is produced. Both siRNA and miRNA work through RNA-induced silencing complex, or risk complex, that can lead to mRNA degradation. Mostly in case of siRNA, they lead to mRNA degradation, or it can also lead to translational block mRNA that usually bind to 3' end of the mRNA and leads to translational block there. So the net result, the downstream effect of these is effect on translation. So translation won't be, there won't be enough translation of that mRNA. Either the mRNA won't be formed, or mRNA won't be translated into protein. And so eventually it will affect the expression. When we talk about functional genomics, that's probably the heavy emphasis of this course is functional genomics, and how to understand the functions of genes. There are two major mechanisms, and we have now talked about both of these: RNA interference, RNAi. RNAi is a post-transcriptional RNA degradation. It's a reversible knockdown, because when we use RNAi in the labs to understand gene function, as most of the time people will use siRNA. So you inject double-stranded RNA or siRNA, and then that will lead to mRNA degradation. So we degrade the mRNA, and then because there won't be any protein, so you will ask what this protein does. So if this protein is absent, or very small amount of this protein is present, then how would that affect this individual? How would this affect the plant? How would that affect an animal? And also you can ask the questions in multiple different ways. Okay, how does that affect the cell itself, or how is it involved in some sort of pathogen interaction? So if you infect the cell after knocking down this protein, how would that affect the viral amplification, for example? Is it important for the viral amplification? So if it is, then a knockdown of that protein will impact the viral cycle. So you can ask these questions by RNA interference. It's easier to carry out than transgenics, because all you need is double-stranded RNA or siRNA complementary to the gene that you want to knock down. So there's also the term knockdown versus knockout, because RNA interference is not absolute. It's not deleting the DNA, the gene from the organism. So it's not a knockout of that gene, rather it's knockdown of the transcript of the expression. It's reversible unless it's under a promoter control, like in Drosophila, that's continuously making these siRNAs and continuously degrading the transcript that we can do in model organism. But in other organism, it will be reversible after a couple of days. These will be the double-stranded RNA or siRNA will be degraded, will be broken down by the cell, and it will be removed. So there is no new siRNA to make the RISC complex to degrade the mRNA. So the effect usually lasts about 7 to 10 days, but it gives you enough time, enough window to see how does that absence of this protein or lower amount of this protein affect the individual. So that's one way to understand functional genomics, to ask the questions, what's the function of this gene? Another way is transgenics, or gene knockout or knock-in. So knock-in means when you insert a gene or you increase the expression of a gene in an organism instead of decreasing the expression, or instead of taking it out from the individual. The transformation of transgenics, gene knockout and knock-ins are more permanent because now we are working on the genomic DNA level. You cannot go back and unless you cross those individuals with wild type multiple times and recover, get rid of that mutation, you will have that permanently in the genome of that individual, the population. For transgenics, we can use a classical transgenic by transposable elements, or you can use the CRISPR for gene editing, either by HDR or NHEJ, which will give you the same answer, what's the function of this gene if you knock out this gene. So if we are looking at these two main mechanisms of understanding functional genomics, of answering what the function of a particular gene, a particular protein is. So RNAi, here you select a target gene, that gene that you want to silence, you want to knock down. Then you design these double-stranded RNA or siRNA, and you can order these just like you would order primers. Once you have these siRNA or double-stranded RNA, you inject these in an organism if you want to see more organismal effect, or in the cells if you're working on a cell line. You can inject, you can transfect the cells, you can electroporate the cells. So either way, what you want is you want this siRNA to enter into the nucleus, into the cell. And then you will detect the knockdown efficiency by real-time PCR or by Western blotting to look if you have antibody available. Then you can see exact amounts of proteins produced in these individuals. In terms of knockout, let's talk about CRISPR here. Then you design your oligos. You design your guide RNA, you assemble your guide RNA and CRISPR RNA. So this is that common sequence that will give the stability, and this is your unique guide RNA for your gene of interest. And then again, you will deliver this guide RNA along with Cas9 by either electroporation, nucleofaction, lipid transfection if you're using cells, or direct injection in the embryo if you are using an individual, an organism. And then you will analyze editing efficiency, usually by Sanger sequencing of that target sequence, to see if there is any differences in the sequence of our non-injected or wild type versus these individuals that you have gene knockout, potential gene knockout. So again, this is RNAi. siRNA will make this complementary base pairing with the target mRNA. The RISC protein complex will form there. This is target mRNA, and this RISC complex has EGO, which is an endonuclease, and this will degrade this siRNA complex, especially this mRNA, because if mRNA is degraded, then it will not be able to make protein. So this is a reversible knockdown. In CRISPR, you have guide RNA. You can inject your guide RNA and Cas9, or you can inject a plasmid that contains both guide RNA and Cas9. and then you will have this Cas9 guide RNA complementary sequence here. This guide RNA sequence will find the complementary sequence in the gene, and you want to make it very specific to your genes to avoid any off-target effects, off-target cutting. And this is here, then this Cas9 comes and bind here. This shows a little too far, but it's only like 2 base pair away from the PAM site. That's where the cut will be. So yeah, well, I guess this is the PAM site. It should show a little further these sequences, but anyway, so it will make this double-stranded break. The Cas9 is the endonuclease that will make this double-stranded break, and then the double-stranded breaks will disrupt the gene. This will be fixed by the cell DNA repair mechanism. So this is all for today, and in the next lecture I will talk about gene expression in systems, a little more about regulation of gene expression, and then I will also spend some time talking about the mechanisms of RDRPs and how it is very important for retroviruses. Okay, with that, well, that's it. Okay, have a good day and be safe.